días a todos. It's my great pleasure to introduce Dr. Marta Biancardi. She completed a PhD in biophysics from the University of Rome. She did 2 postdoctoral fellowships, one at the Foundation Santa Lucia at Neuroimaging Laboratory Rome in Italy, working on multimodal imaging, and a second one at Advanced MRI Section at NIH in Bethesda, Maryland, USA, working with ultra-high field imaging. She is currently an assistant professor in radiology at Harvard Medical School and Massachusetts General Hospital, where she is currently the director of the Brainstem Imaging Lab. Besides this, she is a great woman and one of the most admirable scientific persons that I know. I hope you can enjoy, and many thanks for coming.
Thank you so much for the introduction, and thank you very much to the organizers. It's a great pleasure to be here. And I visited Mexico many times for pleasure, and it's the first time I do it for work. And it's great to know that there is this big neuroimaging community. It's really a pleasure to know about this. And so today I will talk mainly about the brainstem, which is a deep brain area that at the base of the brain. It's a stem-like area at the base of the brain, which can be subdivided into 3 gross anatomical subregions, which are the midbrain, the pons, and the medulla. In humans, it's slightly bigger than a thumb, but it contains more than 170 gray matter regions that are called nuclei. And if you count all the substructures, you reach at least 300 subnuclei. So these brainstem nuclei are involved in several vital functions, such as sleep and wakefulness, autonomic pain and limbic function, sensory function, as well as motor function. And impairment in the structure and function of these nuclei is associated with several disorders, including sleep disorders, disorders of consciousness, autonomic impairments, chronic pain, anxiety, depression, sensory disorders such as vestibular disorders or visual and auditory impairments, and movement disorders. If we zoom in at, for example, at the level of the pons, you can appreciate how rich of anatomical information this area is. So all these are nuclei interspersed with the white matter fiber bundles. And we know this mainly from post-mortem atlases of brainstem specimens, which are detailed, they give detailed maps of the cytoarchitecture and myoarchitectures of these structures. However, in vivo, brainstem nuclei identification, it's more difficult. And this is because of the small brainstem nuclei size. However, I think at least 1/3 of them have a resolution of at least 1 millimeter linear resolution, so they can in principle be mapped. However, the main problem is the limited contrast. So as you can see here, the gray-white matter contrast in the brainstem is much lower than the gray-white matter contrast in the cortex. And so again, if we zoom in at that level in an image, in a conventional image, like a T1 weighted image, this is a template, an MNI template, you can see how flat the contrast is. And basically, brainstem nuclei identification is mainly guided by extrapolations from ex vivo atlases. And some of the mostly are visual extrapolations, or sometimes they're extrapolations based with measuring from landmarks, the distance from landmarks. And this limits the precision in brainstem nuclei identification in research and clinical studies. For example, in implantation of deep brain stimulation electrode in Parkinson's disease. So here we really want to know if we are targeting the pedunculopontine nucleus or the cuneiform nucleus. Or for example, to evaluate microbleed location in traumatic coma patients. So it's difficult to predict if this person will wake up from coma if we don't know which arousal nuclei are affected and also how much. So in general, in vivo human brainstem imaging lags behind the cortical imaging. There was a nice review a couple of years ago that estimated that only 7% of the subcortex has been mapped in vivo. And for example, if you look at structural atlases, and the connectomes that are derived from these atlases. You can see that the cortex is subdivided in more than 100 regions, and instead the brainstem is assumed to be a big lump of all the gray matter nuclei and the white matter. And in this cortical representation, this connectome representation, the brainstem is just represented just by one node, the green node. So as I mentioned before, the issues are the small size of brainstem nuclei, so we need high spatial resolution, the difficulty in localization due to the low contrast in this area. So having an in vivo brainstem atlas, it would be great rather than ex vivo atlases. And then, for example, for fMRI, we have a high level of physiological noise, especially in the CSF surrounding the brainstem. We have a lot of pulsatility effects, and so we need the brainstem-specific processing pipelines. And last but not the least, we have signal dropout and distortions, which mainly affect the anterior pons, mainly this area. These brainstem nuclei are small, so we really need to work a lot on co-registration routines. And so our goal was to develop the brainstem navigator, basically an atlas, a functional connectome, a structural connectome of brainstem nuclei, as well as to evaluate the translatability of 7 Tesla connectivity results to 3 Tesla. So starting with the atlas, the previous work showed that by using multi-contrast imaging, including T1-weighted, T2-weighted, quantitative susceptibility mapping, and magnetization transfer, you can map maybe less than 10 brainstem nuclei. So we moved this forward, and we acquired multiple contrast, and we especially made use of the T2-weighted image and fractional anisotropy at 7 Tesla, and this was a 1.1 millimeter isotropic resolution to delineate brainstem nuclei on single subjects. And then we computed the overlap across multiple subjects across a population to yield a probabilistic atlas. I remember when I started doing diffusion imaging at 7 Tesla, it was not, people were asking me, why are you doing this? I mean, it's, you expect the same contrast as a 3 Tesla. Diffusion properties are independent of the field strength. So it took me some time to convince people and myself that you have a higher sensitivity, so you can increase the resolution. And this means also gaining contrast because also you reduce partial volume effects. And I remember the first time we looked at the fractional isotopy at 7 Tesla, these values, the values in the white matter were higher than values in than at 3 Tesla, slightly higher. And I think this is just due because of the partial volume, the reduced partial volume effects. And so we focused on developing an atlas of 31 nuclei that I'm listing here, and are involved in different functions, and some of them have multiple functions. There is a substantial overlap of function for brainstem nuclei. For example, the substantia nigra and the peduncle tegmentas nucleus and the subceruleus are both involved in sleep, arousal, and in motor function. The solitary nucleus is both an autonomic nucleus and a sensory nucleus. Reticular formation nuclei are very intriguing because they have at least 3 functions. And we computed a probabilistic atlas, so in living humans, of these nuclei. And I'm showing you here 16 brainstem nuclei relevant for arousal and motor functions, and here 15 brainstem nuclei relevant for autonomic, limbic, vestibular, and sensory processing. And so this work was performed by Guadalupe Garcia Gomar and Kavita Singh, who were postdocs in my lab, and by myself, and it took 6 years. I started the first 2 years by myself, and then they joined, and it was an additional 4 years. And I think the pandemic helped us because we couldn't acquire data, so we were just focusing on analyzing and segmenting these regions. And it led to these 6 publications that I'm listing here. And don't worry, I'll describe more these nuclei. It's a lot. So how did we do this atlas? We acquired on 12 subjects 1.1 millimeter T2-weighted and diffusion imaging, and we computed the fractional anisotropy. And this is a single subject image, so we could observe nuclei at the single subject level. And we worked so the key was to have a short echo time, because so you have already less sensitivity in the brainstem because it's farther away from your receiver ray coils. And also I learned that the T2 of the brainstem is shorter than the T2 in the cortex. So you really need to push the echo time to minimize the echo time. And yeah, it took like several months just to understand which was the best sequence and how to make it work for the brainstem. And we performed either manual segmentation or semi-automatic. At first, we were doing semi-automatic segmentations, which I'm showing you here. So for example, this is the original image. Here we performed unsupervised k-means clustering, and then we kind of identified the cluster of interest, which however contains 3 nuclei, 1, 2, and 3. So then we had to perform some manual segmentation, like of these bridges with and just of the bridges, and then we would get automatically these 3 nuclei, and which, for example, this is the inferior olive. So we would repeat this for all the subjects, and then we would co-register them to MNI space and compute the probabilistic label across subjects. And we validated the atlas in several ways. So, for example, sometimes we had the 2 manual delineations, or we had the 1 semi-automatic and then another manual rater. So we had 2 independent people segmenting these regions. And we compared the inter-rater agreement, and we used the modified Hausdorff distance, which is shown here, because it's a distance that it's an overlap measure that's independent on the nuclei size. For example, if you use the dice coefficient or the Jaccard index, they perform better for larger nuclei, and you get a much smaller value for smaller nuclei. And the Hausdorff distance is basically independent on the nuclei size. And for the Hausdorff distance, you take so you have 2 labels. One is the green, one is the blue. You take for each point the minimum distance to the other label, and then you compute the mean across all the green points, and then you do the same for the blue, and then you take the maximum. So it's modified because we take the maximum rather than the mean, so it's a little bit more conservative. And then we say that the atlas is validated if the Hausdorff distance is less than the imaging resolution. So if the imaging resolution was 1.1, this indicated a good performance of the manual delineations. And also we computed the internal consistency. So if you have just one label, you compare it to the mean across the other subjects. So we do a leave one out approach, and we again compute the Hausdorff distance, and we compare it to the imaging resolution. Another form of validation, which we introduced kind of later, was to perform the agreement of label volumes with gold standard volumes. So it was difficult to identify the gold standard volumes because there is not literature on all these nuclei in vivo. And so we used an ex vivo atlas, the Paxinos atlas, which is a 2D, it's a stack of 2D images. So we segmented these nuclei in the atlas, in the ex vivo atlas, and then we computed the volume as the sum of the volumes of individual slices. It's an approximation, but then we compared it to our measures, and we got most of the time good agreement. When we didn't, we either kept going with the delineation or we tried to interpret the results. For example, this nucleus, we improved it in a later delineation with the Guadalupe because we were missing the caudal part, and we worked on it, and we finally reached an agreement. Another form of validation, which was more time-consuming, was histology. And so we performed it only for some nuclei. For example, here I'm showing you the lateral parabrachial and medial parabrachial. We performed Gallia staining of myelin and Nissl staining of cell bodies. And we validated the location, and sometimes also the properties matched with our contrast, as I'll show you for other nuclei. So just to go over in more detail on these nuclei, the substantia nigra and the red nucleus are probably one of the most studied brainstem nuclei. They are in the midbrain, so in the upper portion of the brainstem, and they are hypointense in both T2 and T2 star weighted MRI, and they are hyperintense in the QSM. And we added 2 cents to this, and basically we automatically delineated the substantia nigra as the regions of hypointensity in T2, but then we observed that in fractional isotropy, we observed 2 regions, one with high fractional isotropy and one with lower fractional isotropy. So we delineated both, and they are compatible with the pars compacta and pars reticulata. I think the upper portion is not fully visible, but yeah, we can see it here. So pars compacta, in line with the histology, was medial to pars reticulata. And also interesting, it's known that pars compacta has longer and thicker dendrites and more densely packed neurons, and this might is in line. I mean, it might underline our contrast of higher fractional anisotropy in pars compacta compared to pars reticulata. And also pars compacta is more highly densely vascularized. I don't know if that adds to the fractional anisotropy as well. So reticular formation nuclei, that's a very interesting group of nuclei. It's called reticular because of the neuropil has a web shape with loosely packed neurons. And it's kind of a column going from the midbrain to the medulla. And originally it was thought just as a big inconspicuous area, but then in the 50s they started identifying nuclei. So they said it's organized in at least 10 nuclei. And we were able to identify several of them, including the mesencephalic reticular formation, which is very important for arousal, for example, isthmic, which is less known, cuneiform, also involved in motor function, the pontine reticular formation, motor also function and arousal, medullary reticular formation, both inferior and superior. And as I mentioned before, yeah, it's an area involved in several functions, including wakefulness, arousal, and the coordination of neighboring nuclei, which are motor, autonomic, and arousal nuclei. That's why its involvement in multiple functions. And Raphe nuclei are mainly serotonergic nuclei, and they are midline nuclei, also running from the midbrain to the medulla. And sorry about that, they're involved in sleep and wakeful arousal and autonomic pain limbic function, and Raphe pallidus and Raphe obscurus also in motor function. So out of 7 nuclei, we were able to identify 6 based also here on the contrast. You can see that they are like areas of hypointensity. And so we were able to identify the caudal rostral linear Raphe, the dorsal Raphe, median Raphe, Raphe magnus, Raphe obscurus, and pallidus. And going forward to these are cholinergic brainstem nuclei. We have 2 large cholinergic centers are the laterodorsal tegmental nucleus and the pedunculo-tegmental nucleus, which is also called pedunculopontine nucleus. And they're involved in arousal, attention, REM sleep, and the pedunculo-tegmental nucleus also in locomotion. And so we identified, for example, the pedunculo-tegmental nucleus was this area of hypointensity in the fractional anisotropy, and the laterodorsal tegmental nucleus was lying just behind the white matter. It had a mixed contrast. It led between the gray matter and the CSF. And the locus ceruleus is probably the most studied nucleus in ex vivo. And also now recently has been studied more and more in vivo. It's a very interesting nucleus because it's very small, very visible, for example, in histology. It's kind of dark. The cell bodies are really big, but it connects, it has direct connections with the cortex. So it has the ability to connect to widespread connectivity with the cortex. And it's involved in arousal, autonomic function, attention, memory, motivation. It's really a key brain area. And in 2006, Sasaki and collaborators showed that by using a T1-weighted fast spin echo technique, you can see this column as a white, it's as a hyper intense area in T1 weighted imaging, and they called it neuromelanin MRI because also the locus ceruleus is rich in neuromelanin, and in presence of iron and copper it has T1 shortening paramagnetic effects, and also there is magnetization transfer due to the presence of protein pool of water. And adopted a different approach, and we identified the locus ceruleus also as a column of hyperintensity but in fractional anisotropy, and we were also able to identify the subceruleus as also this column of fractional anisotropy. The subceruleus, it's just anterior to the locus ceruleus and goes down to this is the superior olive. And the subceruleus, for example, is important in limbic function and in REM sleep muscle atonia, and I will talk more in my next talk also about the disorders of REM sleep atonia. And this is, for example, the lateral and medial parabrachial nuclei, and like the lateral parabrachial is lateral to the superior cerebellar peduncle, and the medial parabrachial is medial to the superior cerebellar peduncle. And I think, yes, their function is, they're very important autonomic centers, so they're involved in pain, respiration, cardiovascular function, food intake, and wakeful arousal. And they're at an interface between medullary viscerosensory regions, such as the solitary nucleus, and forebrain autonomic centers. They're really an important relay mediator of this function. And I think this is the last slide about brainstem nuclei. So these are vestibular nuclei. The vestibular nuclei are important for balance. And there are 4 vestibular nuclei, the inferior, superior, medial, and lateral. We were just able to identify a cluster of all of them. And here, just to conclude, the viscerosensory motor nucleus, we gave this name because we couldn't identify in individual nuclei, but it comprises 3 nuclei. It comprises the solitary nucleus, the vagus nerve nucleus, and the hypoglossal nucleus, and they are involved in viscerosensory motor function. And we identified them as being posterior to the inferior olivary nucleus, medial to the medullary reticular formation. And it's interesting that this region is gray matter, but it's kind of white, grayish, and it's because the solitary nucleus surrounds the solitary tract, and the vagus nerve nucleus also is exactly where the vagus nerve is. So it's a mix of white and gray matter, and so the contrast is slightly different than the other gray matter nuclei. And so we recently released, we packed all these 31 nuclei, and we released them in the Brainstem Navigator toolkit, which can be downloaded at that link. To go to that link, you can just go on Google and write Brainstem Navigator, and then it's free, available for use. And so here the video is not running. But it's okay. You should have seen just a 3D rendering from different points of view. The Brainstem Nuclei Atlas of, the toolkit includes, so the Brainstem Nuclei Atlas of 31 brainstem nuclei, and also we have 3 diencephalic nuclei. It was achieved in living humans, specifically in younger adults, as opposed to post-mortem atlases. It's 3D, also as opposed to post-mortem atlases, which are mainly 2D, and it's a stack of 2D images. It's probabilistic, so it takes into account variability across the population. And it's deformable. Deformable means that you can automatically map it to conventional images of living humans by running co-registration of the atlas to the conventional images. And we are now extending the atlas to elder adults. In aging, there are several morphological changes. There is atrophy, there is the increased size of the ventricles, and for example, the position and the shape, these nuclei are pushed compared to younger adults. Also, there is iron deposition. We are still looking if this, for example, iron-rich areas become just bigger or just their contrast is lower. We're still working on this piece. And we hope to be able to release the atlas also in elderly population. Okay, so now I reached the functional connectome of brainstem nuclei, and it was our second goal. And we used in vivo high-resolution 7 Tesla fMRI and brainstem-specific physiological noise correction and co-registration. Yeah, so brainstem hodology indicates that there is an intricate wiring diagram of the brainstem. There is strong intraconnectivity And there is connectivity to the cortex, to the subcortex, specifically to thalamus, hypothalamus, cerebellum, hippocampus, basal forebrain, amygdala, basal ganglia. And also, it's a crucial region for motor and sensory pathways from the cortex to the spinal cord. There are really large fiber bundles traversing the brainstem. And so this intricate wiring diagram can be split based on the function. Here I'm showing you the motor and sensory circuit, the visual and auditory one, the respiratory circuits, the noradrenergic and adrenergic centers and circuits, the autonomic circuits, and the vestibular ocular circuits. And all these were studied and identified in animals. And our goal was to see how they look like in humans, at least to give a preliminary glance at these circuits. And so here, as for the atlas, we split these nuclei into two works. One we focused on sleep, wakeful arousal, and motor function, and on the other work we looked at autonomic pain and limbic function and sensory function. And we achieved the connectomes using 1.1 millimeter 7 Tesla fMRI in humans. This work was mainly led by Kavita Singh and Simone Cauzzo in my lab. And in red, I'm indicating the seeds. So in this work, the seeds were sleep and motor nuclei, and the targets were the autonomic and sensory nuclei, as well as other subcortical areas and the cortex. This is like occipital, parietal, temporal, frontal cortex. And this was the opposite. So these were the seeds and these other were the targets. And yeah, after looking at this, you might think, oh, I prefer the animal circuits. But let's go and see how we derived them and what we learned from them. So on 20 subjects, we acquired 1.1 millimeter isotopic resolution fMRI. We did 3 runs of 10 minutes. So the acquisition time was 30 minutes. I did a preliminary study several years ago just with 10 minutes, and we didn't achieve a great sensitivity. So this was a great boost in sensitivity, which goes with the square root of time, acquisition time. And we also acquired an MP-RAGE for parcellation and for co-registration purposes. And the processing pipeline, I would say it was mainly standard, except for the fact that it was a huge, Guadalupe says, no, it wasn't standard. That was for huge data, big data. And then also we customized the physiological noise correction. We used regressors that were derived from the CSF neighboring the brainstem. We used the 5 principal component regressors from there. We didn't apply spatial smoothing for when we computed the average time course in the seeds, because some seeds lay near the CSF, so they would get all the pulsatility from the CSF, even though we tried to correct for that. Yeah, just to show you this in a picture. So in red and yellow, I'm showing you the areas that show, this is the standard deviation across time of the fMRI time course. So it's very high in areas neighboring the brainstem. And we derived the 5 PCA regressors from there, and we didn't apply the smoothing. And here I have a video, I hope it works. Yeah, you can see it. Yeah, so this is what I'm talking about. So this is the pulsatility effect. This is a cardiac-gated flash image. And you see that there are specific, so this is the aqueduct and the periaqueductal gray. It's a gray matter nucleus. It's around this. So you can see that the pulsatility affects also the nucleus a little bit. This is the 4th ventricle. I think the circle of Willis is somewhere here in front of the VTA. We have also these big basilar arteries or veins, because they are dark, so I think they're more vein. And you can see that there is kind of an antiphase with the lateral. They're not exactly in phase, there is a delay. So having a specific, CSF-specific physiological noise correction near the brainstem improves the correction of these pulsatility effects. And we also worked a lot on co-registration. And I will talk more about co-registration in my second talk tomorrow. But we work a lot on creating a mask. I specify here, we we crop the inferior slices, we do betting, we do manual editing, and also we work a lot on these pieces, on how many iterations and levels and so on. But this is just an example of the quality. We got good co-registration in the brainstem, except for some subjects in the anterior pons, where you can see that the signal drop out. Here we cannot recover, even after, distortion correction, we can recover for distortions, but these are still here, these effects. And in any case, it's mainly the anterior part of the pons that is affected, and all our nuclei are in the tegmentum, except for some nuclei that are here in the anterior midbrain, but not in the pons. And we always check the alignment of brainstem nuclei with our images. So this is, for example, the alignment with the ampérage. This is a 3 Tesla fMRI. This is a 7 Tesla fMRI. And so we use the seeds, the 31 brainstem nuclei labels from our atlas, and as targets the free surface parcellation labels, and we computed the average time course within each seed and target, and then we computed the Pearson correlation value across them. So it's a very simple computation, and this is the connectivity matrix that we obtained. And in another form, this is the two circular connectome that it's just saying the same as the connectivity matrix in a different form. I think it's more useful when you look at the connectivity from individual nuclei rather than at all of them together. And we also computed the voxel-based connectivity map. And as you can see, we achieved a very high statistical threshold. It was a high Bonferroni corrected threshold. In red, I'm showing you the seeds, and in black, these are brainstem targets, and subcortical and cortical targets. As you can see, we achieved high connectivity with the cortex, also in the brainstem, but it was weaker. It was stronger in the cortex. So this was the first observation, and then we observed specifically high connectivity to frontal cortices, subcortical regions, as expected with the literature, and we observed weak connectivity to the visual cortex. And just to give you an example of a couple of nuclei, so this is, for example, the connectome of the locus ceruleus, and it showed expected connectivity with the dorsal raphe, lateral dorsal tegmental nucleus, and these are all arousal areas, the VTA and medial parabrachial autonomic centers, which is in line with the function of locus ceruleus. And also interesting, the thalamus, as expected, the hypothalamus, subthalamic nucleus. The connection with the amygdala, it's there, but it's weak. It's a finding. We don't get good connectivity to the amygdala. Also, we had cerebellum, pallidum, and also the frontotemporal cortex in line with the literature. This is the vestibular nuclear complex, very dense. But if we looked just at the top, the strongest connections, we got the inferior, the opercular and the insula, which are vestibular cortices. They have been identified, I think, 15 years ago, and they are vestibular cortices. And also we found connectivity to the precuneus and the posterior dorsal part of the cingulate cortex in line with the literature. And also we got connectivity with the viscerosensory motor nucleus, but only contralateral. The medial parabrachial nucleus connected with the dorsal raphe and thalamus. Interestingly, it connected also to the cingulate cortex and the insular cortex, and from the literature there is no direct connectivity, but interestingly the thalamus is a relay to this, to the granular insular cortex and the cingulate, so we believe this is indirect connectivity, because unfortunately with fMRI, area A and B can be connected, either because they are directly connected or because they just are connected to area C, and it's an artifact, unfortunately. And yeah, also the PAG and VTA, which were found also in rats. And we found weaker connectivity again with the amygdala and VSM, and no connectivity with the raphe magnus. So we've got expected findings, but also unexpected findings based on animal literature. So to summarize, we, from a system neuroscience perspective, we tested the hypothesis if brainstem nuclei are highly interconnected within each network. And so this is, for example, and we built connectivity diagrams, and this is the network for the arousal circuit, and you can see that, yes, they are highly interconnected. We were very glad to see this, and this is for the motor circuit. Here on top is for the autonomic limbic and nociceptive circuit, and on the bottom for the vestibular circuit. And then we were also eager to see the internetwork connectivity. So we expected a weaker connectivity. So we found actually a sparser connectivity, but it was still there. And for example, this is an important finding for vestibular disorders, because it's known that like anxiety and the autonomic function is important for the proper compensation after vestibular events. So they have been postulating connections between insula and the vestibular cortex and or the infralimbic cortex, and it was good to see this in humans. So now going to the structural connectome of the same brainstem nuclei, we used in vivo high-resolution diffusion imaging at 7 Tesla, and again we worked a lot on co-registration in brainstem areas. And so here we started from a prediction model. So let's see what we have in the literature, and this was derived from Olszewski and Baxter. And so, for example, for arousal nuclei shown on the left in blue, we expect a connectivity to brainstem, thalamus, hypothalamus, basal forebrain, and cortex. Instead, for motor nuclei, which are shown here on the right, we expect connectivity to the brainstem, basal ganglia, cerebellum, and cortex. And I'm also showing you in pink and purple colors. These are nuclei that are involved both in arousal and in motor function, and you can see that connect to both of them, and have some of the characteristics of both circuits. And then we asked, what can we achieve with tractography in living humans? And here we achieved these connectomes with 1.7 millimeter diffusion MRI. So we kept going with diffusion at 7 Tesla, and this work was mainly led by Guadalupe Garcia-Gomar and Kavita Singh, and a similar approach and similar representation. And so here we employed the same subject as for the functional connectome generation. So they participated in both fMRI and diffusion, and was important to decrease so the voxel size was 1.7 millimeter resolution. Also here we used the minimum echo time, and to do this we used the unipolar diffusion encoding rather than bipolar diffusion encoding, and the b-value of 2500. And we also acquired an MP-range here mainly for parcellation purposes. And again, the processing pipeline was with some standard, with some improvements. We did some adjustments to the seed-based probabilistic tractography. We used constraints finally called deconvolution. With 100,000 streamlines per seed. We shortened the minimum streamline length. I think the default was 5. We shortened it to 1 millimeter because some of these nuclei are neighboring to each other and they are known to have connections. We also adopted a less conservative approach for the angle between successive steps. Here we used 90 degrees. And again, we worked a lot on co-registrations. Here, the co-registration, I find it easier for diffusion than for fMRI because you don't go through the MP-range. Here, you do a co-registration from FA and T2A, they call it S0, directly to FA and S0, and I will talk more about this tomorrow. But for example, to show you the co-registration quality, it was basically good in all the subjects, both in the tegmentum and also in the anterior pons. And again, we use the same seeds and targets as for our functional study. And we built similarly a 2D connectivity matrix, a 2D connectome, and a voxel-based streamline density map. So first of all, we achieved less sensitivity than fMRI. We could only use a false discovery rate correction rather than a Bonferroni correction, which is more conservative. The second finding is that the connectivity was much denser and stronger in the brainstem than in the cortex. And I would say the third finding was, yeah, we observed also contralateral connectivity. I remember a reviewer asked us about it. And also we observed connectivity streamlines in the corpus callosum. And this is not so clear, I mean, from the literature if they should be there or not. We can have decussation at the level of the brainstem, which could lead to streamlines also in contralateral regions. Or we could think of introducing, like filtering the tractography in the future to see how if we can correct for this effect. I'm sorry, the figure here is not visible. You should see here the connectivity of the locus ceruleus, but at least we can see here. So it's connected to areas expected based on its function, its arousal function, and yeah, and also here we built connectivity summary diagrams, and we observed a really very strong intra-brainstem connectivity, which we believe it's partially a good result and partially is led by biases, because in the brainstem we have really large fiber bundles, and unfortunately the most tractography approaches there is a bias towards larger fiber bundles. The smaller fiber bundles are less represented, also from a probabilistic point of view. And also the other bias in tractography is that regions closer to the seed are more represented. There is a bias for regions closer to the seed. And we were very curious to compare the structural and functional connectomes, and probably we'll hear more in the next talk. And so if this was our functional results, these are our structural results, and we obtained a significant correlation of the connectivity matrices. And you can see that, for example, in the tractography in the lower, here we have higher connectivity within the brainstem. Here it's more with the cortex, but we observed similar strong connectivity with the frontal areas, with the subcortex, and weaker connectivity with the visual cortices. And also you can see here from the locus ceruleus, you can see that the cortex has more denser connectivity in fMRI than the structural, and instead the structural based connectivity is denser in the brainstem than the functional connectivity. And so in summary, we show the feasibility of mapping functional and structural connectomes of brainstem nuclei by high spatial resolution 7 Tesla MRI. And we identified several connectivity pathways in living humans that are in line with animal studies. And of course, we also found lacking or unexpected connectivity, so the famous false positive and false negatives. Although, I mean, there is not a ground truth because we compare to animal studies, so we don't know if what we found it's a finding for interest for humans, specific for humans, or if it's an artifact. So, yeah, for example, we couldn't find good connectivity with the amygdala for fMRI, and maybe this is due to the dropouts and in these regions, and also to the presence of lots of arteries and veins in that area. For example, we found contralateral connectivity in tractography, and also we found indirect connectivity. I was mentioning one example before, and this is another example. We found connectivity of VSM and accumbens, which should be via the parabrachial nuclei, but we also found direct connectivity. And so just to summarize also the limitations of fMRI, so the Pearson correlation is unable to distinguish between direct and indirect connectivity, so it's called polysynaptic connectivity. There is also residual motion and physiological noise. We have residual signal dropout and distortions. Diffusion-based tractography also has a polysynaptic nature of tractography reconstructions unless you really filter your results, but you also need to have a good prior knowledge of how to filter the results, which it's not easy, especially for the brainstem. There are crossing fibers, There are biases for larger fiber bundles and also for fibers closer to the seed. And so further work is needed to improve the resemblance. We find it like it could be a validation way. If the function and structural connectivity metrics look more like each other, maybe we're going in towards the right direction. So we plan to adopt improved strategy for both tractography and fMRI and see if they look more close to each other. And yeah, now going to the last part of the talk. So we evaluated the translatability of our 7 Tesla connectivity results to 3 Tesla. And on the same subjects, we also did 3 Tesla, for example, fMRI. We just did standard a 9-minute resting state run and 2.5 millimeter spatial isotropic resolution. And for diffusion, 9 minutes and 2.5 millimeter spatial isotropic resolution. And so this is for fMRI. So for fMRI, we got a nice association between the 7 Tesla connectivity results and the 3 Tesla. And then if we counted the common links between 7 Tesla and 3 Tesla for different thresholds, so these are different thresholds, what we noticed was that at 3 Tesla, we retain 95% of the links that we achieved at 7 Tesla, and these are links brainstem to cortex links. Instead, if we look at brainstem to brainstem links, we retained 70% of them. So it was a good number, but we need to know that we lose 30% of the links, or 30% of the links are different at 3 Tesla compared to 7 Tesla. And also you can see it here that, in the cortex, they look more similar than in the brainstem. The 3 Tesla, it's a little bit sparser. And also looking at the connectivity matrices for the arousal networks and the motor network at the bottom, you can see that going from 7 Tesla to 3 Tesla, you mainly lose here, but this is the cortex and subcortex, it's more similar, you retain more. And so this was for the functional MRI, for the structural connectivity. Also here we got good association values, but not as good as for fMRI. And if we counted the number of common links, we got the opposite results. So we were able to achieve 95% of the links at 3 Tesla that were present also at 7 Tesla, brainstem to cortex, this is brainstem to brainstem, and only 75% of brainstem to cortex. And we believe that this is due to the biases. So fMRI seems to have a higher sensitivity in the cortex because there are larger areas, the time courses are less noisier, and also maybe we're closer to the receiver recoil. And the opposite in tractography, we're more sensitive in the brainstem, maybe because of these larger fibers, they're closer to the seed, and it was interesting to see this result. So in conclusion, we show the feasibility of mapping an atlas and the functional structural connectomes of brainstem nuclei by high spatial resolution 7 Tesla MRI. Our results show good translatability of connectomes to conventional imaging. We hope this atlas and connectome could be useful to investigate the brainstem structure, function, and connectivity in health and disease. And we are recruiting a postdoctoral fellow in brainstem connectomics. If you're interested, just get in touch with me at this workshop or via email. And I would like to thank all the co-authors of this work, the funding sources. This is a view from the Museum of Science of the Charles River, unfortunately not from my office. And thank you so much for your attention and for being here.
Thanks a lot for your talk, and I don't know if you have any questions about the talk. Nobody? Okay. Okay, perfect. She goes. Gracias.
Hello? Hola. Hello. Hi. I was just curious if you have, for your atlas, did you have, I don't know, any other ideas of what possible contrast or techniques could help you to improve the A class, besides the one that you actually have already explored?
The contrast for the Atlas generation.
Do you have any other ideas in mind? You know?
Yeah. So definitely, I think we would like to add magnetization transfer, like for the neuromelanin MRI to visualize the locus ceruleus better and the nigra and possibly other regions. We are also collaborating with Marco Palombo, who developed SANDI. It's an MRI-based technique to measure directly the density of cell bodies and also the size. And we are trying to applying it to brainstem and validating with the histology. And because, for example, some nuclei have a higher cell body densities than others, and so this could be a way of achieving contrast across these regions. We really would like to go to also smaller regions, so to increase the spatial resolution, and maybe look also at other diffusion. I think diffusion-based contrasts, other tensor invariants or other diffusion-based contrasts could be useful.
Maybe, I don't know if you have seen this, the contrast that they generate with oscillant gradient encoding that usually are more sensitive to cell sizes. So maybe it can, maybe it can go there, something, I don't know.
That would be amazing.
Yeah.
That would be amazing. And also we're doing another validation with microCT. We are trying that too, because with microCT, you can look at chunks of tissue, but it's 3D. Maybe you can do 2 centimeters, so rather than having the histology slices, you have a 3D form of validation. But that is also some work because it's new. Yeah.
I missed this. The DTI was, sorry, the tractography was DTI tractography, right?
Yes.
Yeah. In the connectivity?
Fusion weighted.
Yeah, but, okay.
Yes, it's not deterministic, it's a probabilistic tractography.
Okay. I also have, are you planning to check the connectivity, diffusion and functional connectivity in the older subjects and compare them to the younger ones? I suppose you show just the connectivity maps from the younger adults, right? Yeah, I suppose they will be different. Or
yes, it would be interesting to go to look at the older population. We kind of have it, but we never formally did the comparison, older versus younger. But it's a very interesting thing to do, definitely.
Okay, thank you.
Thank you.
What about task fMRI? Will you be able to detect changes in signal in brainstem nuclei?
Yeah, it's a very good question, and it's very difficult to get task-based fMRI in the brainstem because of physiological noise, motion, and also the decreased sensitivity. We just run one task-based study, which is a caloric vestibular stimulation, which induces nystagmus, and because it's the first study done in humans, and we're still analyzing the data. But it would be also validation for our atlas that the vestibular nuclei are active during this task and to see their connectivity to the vestibular cortices. And I collaborate with other good collaborators that have done task-based studies of fear to study the PAG. And we could see activation in sub-regions of the PAG that were in line with the expected location of these activations. And I have another collaborator who's doing meditation, and he's looking at, it's a meditative task, a series of meditative tasks. And so it's all a work in progress. We don't have results yet.
Okay, thank you.
Thank you.
Hi. You said that you did you did two connectivity atlases, one with 3 Teslas and the other with 7 Teslas. That's correct?
Yes.
My question is about the coils, the antennas. The antennas that you put into the patient, where are they? Because I have understood that the coils that are on the head, on the front of, yeah, don't receive that much of a signal. And I think that Guadalupe once told me that there are some antennas that are put into the brainstem directly. So this is the first question. The antennas that you used in both atlases are from the head or in the brainstem directly?
It's a good question, and it's not obvious because the brainstem is just the continuation of the spinal cord, and so basically we use head and neck coils, and we use the 64-channel coils. They're head and neck, and also they are custom made. So, for example, at 7 Tesla, the coil usually the coils just follow straight, and instead these bends a little bit, so you achieve higher sensitivity because it's closer to the brainstem. And we also did so at 3 Tesla. A reviewer actually asked us, "Oh, you say it's conventional imaging, but how much would that have impacted?" I mean, there are Nova coils at 3 Tesla, the 64-channel coils. So, and I think we compared the sensitivity, and in the brainstem was slightly higher, but this coil was particularly, it gave improved sensitivity in the cortex. So we were good. We could say it was almost conventional.
And to what extent does using these both antennas compare with using only one side antennas, in the cortex? Do you know how much the signal improves?
Yeah, so I don't have quantitative numbers, but I remember we did an experiment at 3 Tesla with a 32-channel coil, and we couldn't achieve good sensitivity, especially in the medulla. Maybe you get the midbrain. The pons, maybe just in some subjects, and the medulla not that good, but also it depends on the subject ability to really go in the coil, so the top of the head should touch the coil. If they have a short neck, maybe it doesn't help. So, and some are reluctant to really go in. So, for example, in older subjects, I remember we ran maybe 20 subjects, and we couldn't use maybe 8 of them. And in younger, it was much better statistics. So it's a combination of factors.
All right. And just a last one, how did you test your registration performances? Because you showed us an image, and it looked like a layout in which you compared your registration with some kind of layout. What layout is that?
Yes, it's just the borders. There is a command in FSL called slices, which does it automatically. So you can use that. So we compare external borders and also internal borders of structures. And then our final co-registration quality check is when we bring the labels into the images. And but that it's a qualitative, because we know where they should be. So, but in some contrast you can see them, because in fractional isotope, in T2 star, for example, you can see the nigra and the red nucleus. So some of them you can check with the contrast. And unfortunately, it's it's kind of a skill that you acquire. The more you look at these images, the more you're able to evaluate the goodness of the fit. You can also look at the residuals, but the residuals are global of the whole image. I don't know if, or one should do just local co-registration and just look at the residual for the
extraction.
Yeah.
All right. Yeah. Thanks.
Well, first of all, congratulations to you and Dr. Guadalupe for all this work. I had only begun to understand how much work is involved, and this is amazing. Congratulations. Thank you. I was wondering about the need, or why you didn't choose to use super resolution techniques, both in the anatomical images and on diffusion weighted images that, for example, track density imaging. This seems to be like one of the problems where these tools could help, and you must have very good reasons why you avoided them.
Yeah, so it's mainly the size of the brainstem nuclei. So you want to reduce partial volumes, and yeah, you want to match as much as possible the anatomy of these nuclei, also for seeding purposes, to get a higher specificity of your results. Otherwise, it's a mix of nuclei, and you don't want that. I don't know if I got the
No, what
question.
I mean is image processing techniques for the to obtain super resolution based on semi-low resolution. Well, your image is already high resolution, but
Yes, the
go further.
density.
Yeah, track density imaging or image contrast techniques that use
Super
high resolution on one side and a different contrast on the other, and you transfer the contrast. I wonder why, what the reasons are for not using these kind of tools.
No, I think there is no reason. I think we should do this. And I remember also Guadalupe being very interested. And definitely, we can look deeper and get information that might not seem visible, but it is through these super resolution methods. I think there is a lot we still have to do. Yeah.
Thank you.
Thank you.
Congratulations again.